Cellular respiration, part 2. Today we're going to talk about the electron transport chain. We're going to talk about fermentation and maybe just a little bit about photosynthesis. So let's do this. Last day, we left off, we talked about glycolysis, we talked about pyruvate oxidation and the citric acid cycle, and I had a minute to kind of just introduce oxidative phosphorylation, and that includes the electron transport chain and chemiosmosis. So just as a reminder, I'll just go over to this whiteboard here. We drew this, what did I just do? Move that. We drew this beautiful picture, and or maybe yours is even more beautiful than mine. But this is kind of our synopsis of everything so far. So we have over here, we have glycolysis, and then we had pyruvate oxidation, and then we had the citric acid cycle. So at this point, just to summarize what we're talking about, we had produced 4 ATP. 2 ATP were produced in glycolysis, 2 ATP were produced in the citric acid cycle. So the last thing that we need to do is make more ATP, and we're going to do that by using these things here, these NADHs and these FADH2s that were created all over the place. We're going to make a whole load of more ATP. So let's take a look at how that is done in the mitochondria. All right, back to the PowerPoint. Here we go. So just kind of a reminder about the mitochondria. I showed you this picture before, and we were talking about all these features of it, such as it has its own DNA, it has its own ribosomes. And so today I want to talk about what is going on inside the mitochondria and what is going on in the inner membranes. So the inner membranes, by the way, I think I mentioned this before, are sometimes called cristae, meaning that they're kind of look like little fingers under electron micrograph, and that's just the folds of those inner membranes. So here's some other pictures of some mitochondria. I think I showed you this one before. I like this one here because it's kind of that hybrid of the cartoon and the electron micrograph, and you can see how the artist has tried to show those structures, the inner membrane and the outer membrane. And the innermost part of the mitochondria is called the matrix. Lots of pictures of mitochondria. I like pictures of mitochondria. They're very cool organelles. So before we go any further, I just wanted to draw one for you and make a few notes about what's going on in a mitochondria. So here's my mitochondria. I drew it. Actually, I traced it, so it turned out really nice. I'm very proud of myself. Anyway, I want to label a few things. First thing to label is this here. This is the outer membrane. You may wish to take some notes as well. So it may take you a little longer to draw a mitochondria. Usually when I draw one on the board, it kind of looks something like this. So I thought I'd make something a little prettier for you today. So the outer membrane, don't have too much to say about that. It has some transporters. Molecules can get through. It's a relatively porous membrane. We also have, I'm going to leave a little bit of space because I have 2 things to label here, but we have the inner membrane. I'll come back to that in a moment. Inner membrane. And this thing here in between is called the intermembrane space. So kind of like international, it's in between things, right? Intermembrane space. So what's going on in the intermembrane space? Here is where we are going to make something called proton motive force, which we're going to talk about that very shortly. We already talked about it a little bit already, and we're going to get back to it shortly. The inner membrane, this is where we have the electron transport chain. So electron transport chain. By the way, from now on, I'm just going to call it the ETC when I write it because it's a lot quicker to write. And we also have the ATP synthase. So we're going to use both of those as part of oxidative phosphorylation to make ATP. Okay, we're running out of space, but that's all right. We have one more feature that we want to mention, and that, of course, is the matrix. And that is the innermost component. So the matrix, this is where you have your DNA. How do I get rid of that? I made a mistake. I meant to I'll just scratch it out and make a better D than that. DNA, and we also have ribosomes. And for respiration, the important part of this is that this is where we have the citric acid cycle. So citric acid cycle. And maybe not as important for Biology 107, but if you're interested, this is also where fatty acids are broken down. And if you take other biology or biochemistry courses, you'll probably talk about beta oxidation, and that is where basically fatty acids are being broken down. But for this course, that is kind of the synopsis and the quick part of the things we're going to be talking about. Okay, so let's move on and talk about oxidative phosphorylation. And I actually have a quick short video for you. I think this is maybe a minute and a half long, which kind of, it's not the best quality video, but it was kind of short and sweet. And I felt like it does a good job of explaining what is going on in a short amount of time. There's lots of great ones on YouTube that are like, 12 minutes long and things like that. This one I thought was one of the better ones, so I'll play that for you, and then I'll explain it myself. So here goes.
Electron transport phosphorylation, the final stage of aerobic respiration, takes place in the inner membrane of the mitochondrion. This membrane is studded with enzymes and electron carriers, arranged in precise order. Electrons donated by the energized coenzymes NADH and FADH2, produced earlier in respiration, are passed along this transport system. Energy liberated by this process is used to create a gradient by accumulating hydrogen ions in the outer compartment of the mitochondrion. The passed electrons are eventually transferred to oxygen and joined by hydrogen ions to form water. Additionally, the energy of the hydrogen ion gradient drives ATP synthesis, a process which accounts for most of the ATP produced during the aerobic respiration of glucose.
In eukaryotic cells containing mitochondria, the complete breakdown of glucose produces 36 to 38 ATP, with all but 2 produced in the mitochondrion.
Okay, so like I said, short and sweet. Maybe you'll have some dramatic music to make it more exciting. But let's talk about it, and let's take a look at the images from the textbook. So here is the electron transport chain. You can see it's broken down into a few parts. That blob on the right isn't labeled yet. That's the chemiosmosis part. But let's talk about the electron transport chain. If you take a look at the electron transport chain, it is made out of, you can see we have 4 complexes, 1, 2, 3, and 4. So these complexes are these big protein ion complexes. And what they do is they take electrons, again, in as part of chemical bonds, and they're going to move them basically from one molecule to another. So if you take a look at the very first part of the electron transport chain, complex 1 is NADH, and takes NADH. So remember NADH, I was drawing it for you before, kind of like this, right? NAD, and then with a bond, and then an H. So that is going to break down, and you are going to get NAD+. You can see that is right here. So that's going to get recycled and go back to glycolysis and pyruvate oxidation, all that. You're also going to get a proton. So proton, by that I mean a hydrogen ion, plus 2 electrons. So the electrons, like I said before, are not free. They are moving along to another compound as a part of a chemical bond. So these arrows here, you can see they're showing these arrows. It's kind of these wavy arrows going along. And there's one in there called Q, there's another one called cytochrome C, and so on. And to, kind of summarize, again, you can read about this in more details in your textbook, but I don't want to go through every single reaction that's in there. There are quite a few. Basically, what is going on is they are changing from one compound to another, and eventually, those electrons end up over here. So what do we have here? We have oxygen. So oxygen is good. So if I wanted to fill out this equation in full, I have 2 hydrogens, 1/2 O2, so really one oxygen atom, and whoever did this, they're just trying to balance the equation, right? And you're also going to have 2 electrons, but the 2 electrons, of course, are coming from this electron transport chain. So to start off at NADH, get transferred to complex 1, get transferred to complex 2, and then, not complex 2, then to Q, then 3, then cytochrome C, then 4, and then eventually eventually oxygen, and we are making this thing here, H2O, which of course is water. So what does this mean? It means that oxygen is our terminal electron acceptor, our final electron acceptor. So this is why we need oxygen. Without oxygen, what's going to happen? You're going to die, right? None of this can happen, right? And so all of this stuff here shuts down, and we can't do this. And without this electron transport chain, as animals, we die. We can't make enough ATP. So why are we doing this? Why are we moving all these electrons over to oxygen to make water? Well, if you take a look at the other part of this formula, we have these hydrogens. So take a look. We're taking these hydrogens. We're taking from inside. We're taking them from the matrix, and we're moving them into the intermembrane space. So we're actually moving a whole bunch of hydrogens. You can see we're moving them at complex 1, 3, and 4. And as a consequence, up here, we're making our proton motive force. Proton motive force. So what is a proton motive force? Proton motive force is chemical, and it is an electrical gradient. I was going to put the word gradient twice, I just don't have room on there, so I apologize for that. So I've made a chemical and an electrical gradient. And so that's great, right? It's kind of like, we're capturing some energy. So, an analogy would be, we talked about that potential energy with the diver, and he climbs the diving board, and he stored up potential energy. So same thing here. We've pushed some chemicals across a membrane, and you probably know from all that stuff we talked about diffusion, how it wants to diffuse and spread out equally. So that's what it's going to do, and that's where the last part of this thing is, this thing I haven't labeled yet, the chemiosmosis part. So first time the electron transport chain is just moving protons across, producing a proton motive force, eventually reducing oxygen and making water. The second part, the chemiosmosis, so I apologize, I've kind of crowded in my writing there a little bit, but you can see this as ATP synthase. This little thing here is ATP synthase. And the hydrogens now have a channel in which to go from the intermembrane space to the matrix. So basically, as that proton motive force is released, it goes through this ATP synthase, and this actually has part of the this thing actually rotates. And as it rotates, you can kind of, I don't know if you can see my video very well, but I think of enzymes as like hands. So they kind of open and close. So in this case here, as it rotates, it opens, it grabs ADP and inorganic phosphate, and as it closes, it pushes them together. So it literally just does this. As it rotates, opens and closes. As it opens, it brings in substrate. As it closes, it physically moves these things together and makes a chemical bond out of it. So the ultimate goal here is we're making ATP, right? And so we're going to make a whole load of this because we've got a whole bunch of NADH. And I think the last thing I have to point out here is I forgot to mention we have that FADH2. So FADH2 right here actually goes in at a different part of the pathway, right? It goes in at complex 2 and also pumps some hydrogen across, just not quite as much, a little less energy in FADH2. So this is kind of a little image showing the ATP synthase, and you can see the little protons are going through and turning this whole thing like a turbine. And so let's just go back and think. We were talking about something before, something I told you before that was powered by the proton motive force and something that also rotates. So maybe you're thinking now, what rotates? We talked about the bacterial flagella. So the bacterial flagella is also powered by the proton motive force. Also powered by the And I'm just going to write PMF, proton motive force. So just something to think about there. So like I said, as this thing, as the protons go through, it allows the catalytic portion to basically make ATP. So this is great. So I think I have another picture or two here. I do. And you can see there it is, the motor part of this turning and the catalytic part sort of opening and closing, allowing ATP to be made. So one more little kind of footnote to this. Notice this note at the bottom. This here can be run in reverse. If we wanted to, say, hydrolyze ATP and pump hydrogen, we could do that. So anything that's acidic, so your lysosome or your actual stomach, this is how it's done. We pump protons to make something acidic just using ATP. So let me just check here. I know I have a little video. Okay. And then what I want to do is kind of just go back and count a few things and figure out how many ATP we're actually getting. So I'll play this for you, and it's going to show the process. Again, this is not the best video, but I found it was kind of a short one and pretty decent. So I'll play this for you right now.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
So I'm going to stop that there. It just kind of goes on with the music and doesn't really say much new to add to that. Okay, so I want to talk about all these ATP that we have, and I'm going to not necessarily redraw this, but what I want to do is kind of do an accounting of where all the ATP have come from. We've already talked about that we're producing 2 ATP in glycolysis and 2 in the citric acid cycle. And apparently about 26 or 28, and I think the other video said something like 38 and all that. So I want to show you where these numbers come from, and there's different ways to count the numbers. But I'll show you one way to count, and I actually come up with the same number as the textbook. At least this edition of the textbook. The previous edition, I think, had 38 on there. So just different people deciding how they want to count the numbers. So I want to show you how we can count the numbers. And I'm just going to move over to my whiteboard. Let's see here, there we go. And I'm going to start a new whiteboard here. You can see I'm calling it Energy Accounting in Respiration. And so this is the kind of thing I don't necessarily need you to remember every particular detail here, but you know, it's good to know kind of where things are coming from and you know, be able to answer questions about, you know, on the test, for example, you know, where more ATP is being made and understanding, you know, for example, that NADH is going to produce ATP and so on. So let's start off here. Let's just write glycolysis. Okay, looks like I have a funny colored pen. Let's just try something else. That's a little bit better. The other one was some sort of rainbow pen. I don't think I'm going to use that today. It's just looking a little weird. Okay, start off with glycolysis. So there we go. So remember from glycolysis that we made 2 ATP, right? So that was one of our products. I'm going to say net because, of course, we did actually make 4, but remember we consumed 2, so it's 2 overall. Okay, and then I'm just going to put it over here, and this is our total so far, so 2 ATP. Okay, and so we also made 2 NADH. So I'm just going to, let me see here, what can I do? Maybe I can make a little side note over here. So note So in the mitochondria, uh-oh, what just happened there? Somehow it shrunk. Must have bumped something. So in the mitochondria, one NADH will produce 2.5 ATP. And one FADH2 produces 1.5 ATP. So you're probably also thinking, why do I have such funny numbers? Why don't we just have whole numbers? You know, why do we have to break everything in half? Well, it turns out there's a bit of a cost, right? If you make an ATP in the mitochondria, now it's stuck in the mitochondria. So there's actually a cost to getting that ATP out of the mitochondria. And that cost is about 0.5 ATP. So sometimes people say 1 NADH makes 3 ATP. I'm saying 2.5 because there's a net cost that's being consumed as you're taking things out of the mitochondria. If you didn't get that, not a big deal. I won't ask you about that. But I'm just showing you the formula that I'm going to use here for getting things and converting everything to converting everything to ATPs. So we can write that here. I'm going to multiply by 2.5. And so 2.5 times 2 is, of course, 5 ATP. Okay, so the next step was, of course, pyruvate oxidation. Okay, I'm just going to expand this a little bit. It's just make it easier to write for me. So we made no ATP there directly, but we did make 2 NADHs. So same thing as before, we're going to multiply by 2.5, and that is going to give us 5 more ATP. Okay, so the third one was the citric acid cycle. Citric acid cycle. Okay, so remember there we made 2 GTP. We didn't actually make ATP, but like I said, we can just convert them directly to ATP. It's energetically equivalent. So I will just write it directly over there as ATP. We made 6 NADH, and we made 2 FADH2s. Okay, so the 6 NADH, we're going to multiply that by 2.5. So I just got to think for a minute. That is 6. Okay, that's 15. And then the FADH2, we're going to multiply by 1.5. So 1.5 times 2 is, of course, 3 ATP. Okay, zoom out just a little bit, because now the last thing we need to do is just add all this up, right? So we've got 5, 2, 12, 14, 29, 32. Always got to double check my numbers at the end. Always a good idea to double check your numbers to make sure that everything works. So I got 32 ATP, which is the same as the textbook. So like I said, sometimes there's different ways to count these numbers. And by the way, like if you're looking at muscle tissue versus liver tissue, sometimes the numbers are a little bit different because of little quirks in the metabolism. But 32 is good because that means I'm agreeing with the textbook. And so that's 2 sources agreeing, and that's always a good thing. So that's where all the ATP is coming from. You can see 4 are just from making ATP directly. And the rest of them, the other 28 ATP, are being made from all of that NADH and all of that FADH2 that we've produced. Okay, so I'm going to go back to the PowerPoint, and we got a couple other things to talk about here. So we have to talk about fermentation, which is relating to last week's lab, so we can't forget to talk about that. And fermentation basically asks the question, what if there is no oxygen? So notice that little window that I put down that says, what if there's no oxygen? Very strategically, I blocked out all of those 3 processes. Now, technically, oxygen is used in the mitochondria, right? Technically, it's used during the electron transport chain. But it turns out if there is no oxygen, everything in the mitochondria gets shut down. So that means we get no pyruvate oxidation, we get no citric acid cycle, and we get no oxidative phosphorylation. So all we get are 2 ATP. I guess that's better than nothing, but I would sure love to have 32. So this is what happens in mammalian cells, by the way. Okay, so if if you're familiar with lactic acid, particularly people who do a lot of training, weight training and things like that, you might be familiar with lactic acid production. So picture this: if you go on the floor and let's say you start doing push-ups, right? And it doesn't matter whether it's 5 or 30 or whatever. Eventually, most of us, after enough push-ups, you're going to feel a burning sensation. Your arms are going to get kind of wobbly, and what's happening there? Two things are actually happening there. One is that as you do your exercise, you're consuming all your oxygen, and the oxygen gets consumed very quickly, and so there's no oxygen, no energy. Secondly, that burning sensation is actually something called lactic acid. So this is lactic acid down here. Actually, it says lactate, but if I add a hydrogen onto there, that's lactic acid. So same compound, just ionized versus not ionized. So what exactly is going on here? Well, if you take a look just at the top of this, right? At the top here, we have basically right here, we have glycolysis, okay? So glycolysis, converts glucose to pyruvate. We know that before, and it makes 2 ATP. So our muscles can continue to make ATP through glycolysis. It's not a lot of ATP, but our body says, hey, do it, do another push-up or whatever. And so we do. We make a tiny bit of ATP. But if we want to continue glycolysis, then we need more NAD+. So the only way to make, in the muscles anyway, to make NAD+ without oxygen is to take the pyruvate and basically make it into lactate. So if you see what's going on here, basically it recycles the NADH to NAD+. So we're losing out on some ATPs, but in the end we can still get a little bit. Unfortunately, that lactic acid does burn. Eventually your arms will get wobbly, you won't be able to do any more push-ups. And so that lactate will have to be sent to the liver and broken down. So you can't go on without oxygen forever, but you can go on for at least a little bit, right? So maybe push yourself a little bit harder. And in some cases, this is important, right? If this is part of some sort of, is going to protect you or allow you to do something that is essential, this is a very important type of process. So this process, of course, is called fermentation. It means we're breaking down glucose, but only just a little bit. We're breaking down pyruvate only just a little bit, and trying to get a little bit more glycolysis going. So what if you're another type of organism? So what I want to do is show you a video here. And this is, why you came to class today, by the way. This is a nature video. It's pretty old school. There's a lot of good old school nature videos. This one is called Animals Are Beautiful People. So I'm going to play this for you, and then we're going to talk about what's going on here. And so I'll just let it go. And like I said, this is why you came to class today. This is one of my favorite videos.
The only excitement in their lives comes in high summer, when the marula trees are in full fruit. At first, the marulas are very tasty and hard to get at. You have to be either agile or strong to get your share. But after a few weeks, the marulas get overripe and they start dropping from the trees. Then all the animals wade in and gorge themselves on the rotting fruit. It starts fermenting in their tummies and turns into a very potent brew. So on the way home, things start happening to them.
So unfortunately, I'm going to stop it there. It does go on for quite some time with some animals staggering around, and it's very funny. You can check it out. I'll have the video linked on my PowerPoint, but very entertaining video. And so what are we talking about? We're talking about alcohol fermentation. right? So, you know, just some other things I'd show you. I got this, somebody emailed me this a while ago, Canadian beer fridge. So we're making ethanol and carbon dioxide, right? And these are products that are very important in the food and beverage industry. And this is, like I said, the topic of last week's lab. This is what yeast do, is they do alcohol fermentation. They make alcohol and carbon dioxide. This is the reaction. So the same reaction as before. You've got glycolysis here is the first part. And the whole idea is to continue to make a little bit of ATP. But in order to do that, we have to have NAD+. So the only way to do that in the yeast, because they have different enzymes, they don't make lactic acid, they make ethanol and carbon dioxide instead. So you can see the first step here is the pyruvate is converted to acetaldehyde, some carbon dioxide is produced. So if you like to make bread or pizza or something like that, we usually put yeast in there, and the carbon dioxide is what allows the bread to rise. If you don't put it in there, which I did do once, I made pizza and forgot to add the yeast, and it was edible, but kind of just barely, unfortunately. It helps the bread out, right? If you're looking at other products like alcoholic beverages, then this ethanol is the important part of that formula. If you're looking at a carbonated alcoholic beverage, such as beer or champagne, then you're doing both. You want carbon dioxide and you want ethanol. Ethanol, of course, has many, many uses. It's used as a biofuel. It's used in hand sanitizers. So a very important industrial kind of biological product. So another thing I was going to say about lactic acid, what use is lactic acid? Actually, lactic acid is found in a lot of foods too, such as yogurts, by the way. Okay, so you can think of pyruvate as a branching point. And if you watch the lab video, I showed you this exact image, and you see pyruvate is produced always. And if there's oxygen, then we can do everything. We can go through pyruvate oxidation, we can do the citric acid cycle, we can do oxidative phosphorylation, and we can make a whole load of ATP. If we don't have oxygen, then we're going to make some other products, some fermentation products, and that kind of just depends on what kind of organism you are. If you're a mammal, it's lactic acid. If you are certain types of microbes, it might be ethanol, and there's other possibilities as well. Of course, not all organisms have mitochondria, so they may not do the mitochondrial or the aerobic respiration route in general. So kind of back to bacteria, you know, as I had talked about before, we've got all of these different types of bacteria with all sorts of different, this is the slide I'm looking for, with all different types of metabolisms. Of course, yeast here is not a bacteria, that's a fungi. But there are bacteria that make ethanol. And you can see all these products, right? Like I said, lactic acid, that's found in yogurt, ethanol in alcohol drinks, propionic acid, acetic acid. Acetic acid, of course, is vinegar. You can see we've got acetone here. We've got acetic acid again. So quite a variety of different products that are used very widely in the food and beverage industry. The other thing I wanted to mention is that you know, I think I showed you this slide before, way back in topic, I think it was topic 3, and I said I'd come back to this slide. And you can see the terminology I have here is the terminal electron acceptor. So what does that mean? That means where those electrons end up eventually. So remember, I just showed you what happens with aerobic respiration, that oxygen is required and ultimately it makes water. So this is us. We are an obligate aerobe, which means we must have oxygen. And without it, we die. So not good. Or in the short term, we might make lactic acid. By the way, that's what happens to us if people get poisoned by cyanide. Cyanide actually affects that cytochrome C protein I showed you in the electron transport chain. And then the electrons can't move on to oxygen. And so basically, when you get poisoned by cyanide, you're suffocating. So you can see I have another group here called obligate anaerobes. So these things are actually poisoned by oxygen, and some of them undergo something called anaerobic respiration. So you can see here it says, for example, they could use sulfur. And some of these weird microorganisms, what will they make? They'll make something similar to water, but H2S instead. And that's not good. That's hydrogen cyanide. That's actually a toxic gas. But for the bacteria, that's just their waste product. And for them, it's a normal thing. And then we have a third group here, facultative anaerobes. This is kind of like E. coli. E. coli can survive with or without oxygen. If there's oxygen, great, it'll make lots of ATP. If it doesn't have lots of oxygen, it'll just do fermentation and make ATP that way. Small organisms, it's not a problem. They don't need millions of ATP molecules per second. They can survive without it. Okay, so a couple other things about respiration to kind of round this up. First is we talked about control of respiration, right? So remember we talked about phosphofructokinase. You can see this is the, I drew you some pictures before. You can see here, they're kind of drawing what I drew and showing that ATP inhibits it. And it is also inhibited by, it looks like it's also inhibited by citrate as well. That I didn't know. I had mentioned it was inhibited by PEP, which is phosphoenol pyruvate. It's stimulated by AMP, it's also stimulated by ADP. So if you're short of ATP, you're gonna have lots of ADP and you're gonna have lots of AMP. So that's the first thing to mention, that this whole thing is regulated. All of this can be regulated by phosphofructokinase based on really your ATP levels and your metabolite levels. Second thing to mention is we don't just eat glucose, right? We eat a whole bunch of other things. Hopefully you have a nice, healthy diet, and it's including proteins and fats as well as your carbohydrates, right? So it turns out that all of these things are broken down catabolically by our body. And you can see here, for example, proteins are broken down into amino acids, and different amino acids can actually get converted to various metabolites along these pathways. Some amino acids are converted to pyruvate right here. Some amino acids are converted to acetyl-CoA. Others are converted to various metabolites in the citric acid cycle. And they can be broken down, and we can get ATP just by breaking down our amino acids. Carbohydrates, like I said, there's many carbohydrates. Some of them enter at the beginning, some of them a little bit later. Fats are getting broken down into glycerol and fatty acids. You can see the glycerol is converted to glyceraldehyde 3-phosphate. That's a 3-carbon sugar. And the fatty acids are converted to acetyl-CoA in the mitochondria, and the acetyl-CoA can get burned, and it's a lot of energy. You can see the last thing here, I've added those red dotted arrows, and these are representing anabolic pathways. So all of these pathways are intertwined in that we can break things down for energy, or we can use energy to build things up. So for example, right, you eat food, and some of it's protein-based, and you're using that to build muscle, right? And so we can construct amino acids, we can construct proteins, and run all these pathways in reverse. If you eat too many calories, you probably know it will get converted into fat tissue. And same thing, so we can make acetyl-CoA, make fat tissue, and store it in our bodies. So, like I said, that's just kind of a little footnote to this whole thing. You take some more biology and biochemistry courses, you're gonna look at these things in way more detail. But keep in mind that we're looking at glycolysis because it's a central pathway, but it's just one of many. Okay, so what I have here is one of these 3-minute videos for you, and then I also have a Kahoots for you. So I'll play this video, and it's kind of just going to go over what we talked about, and then I have a Kahoot for you that we'll play and see how much you can sort out from the Kahoot. So here goes.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
So hopefully everybody's brain cells are powered right now because we're going to try Kahoots. So you can load up the app, and I will, where is it? There it is. So get ready to join. The number should be loading up there in a second. So I'll give you about 30 seconds to join in. Okay, I'll start this in about 10 seconds, and you can always join at any time. The pin will be at the bottom of the screen. Okay, let's give these a try. I think I got 8 or 10 questions. Some of them are easy, some of them are not easy. Question 1, glycolysis requires oxygen. False. So glycolysis, remember, is the very first reaction pathway, and it happens outside of the mitochondria, no oxygen required. Question 2, where does glycolysis occur? So the correct answer is the cytoplasm. So glycolysis has nothing to do with the mitochondria, so not good. 0 people got that one right. Okay, so eat your food, power those brain cells. Okay, question 3. Told you they're not going to get easier on this one. The oxygen that is needed for cellular respiration is needed for which process? Where exactly is the oxygen required? Better not answer glycolysis. Okay, oxygen is required at the electron transport chain. Okay, so I'm sorry I programmed this not very well. I made this really quickly. I would have also accepted number 5, oxidative phosphorylation. I guess I had 2 right answers, and it programmed it to say answer. The red one was correct. So if you guessed the green one, good job. You're just not going to get the points today for it. Sorry about that. Okay, question 4. ATP is one of the products of glycolysis. What are the other 2? Okay, remember glycolysis. We're not making carbon dioxide. Okay, you know what happened here? And I really apologize for this. I copied questions and then didn't change which was the right answer. Correct answer is the blue one, NADH and pyruvate. It just forgot to change the correct answer on this. So good job, everybody. Most of you got that one right, correct. So hopefully I don't have too many other errors in there. Like I said, I made this really quickly at like 3:51 this afternoon. All right, in the absence of oxygen, yeast cells undergo fermentation, which makes what? Remember yeast, we use them in lab 6. So the correct answer is ethanol. Okay, remember yeast make ethanol for wine and beer and carbon dioxide for bread products. So the answer should have carbon dioxide and ethanol in it. Okay, so a little bit of studying needs to be done here. Question 6, during stage 2 of cellular respiration, acetyl-CoA enters what to form the ATP? So the correct answer is the citric acid cycle. So a little bit better there. That's correct. That's the next step. Okay, a couple more here for you. Which step of cellular respiration generates the most ATP? Hopefully this one is fresh in your head. That one was fast, 10 seconds. Everyone who answered got it correct. Well done. Question 8, what type of cells can undergo fermentation? Prokaryotes, eukaryotes, or both? And the correct answer is both. Remember, our cells can undergo lactic acid fermentation. Microorganisms can undergo a variety of fermentation. So the correct answer is both. Okay, looking hot, Aladdin. 2 more questions. The production of yogurts and some cheeses are the result of what process? This was not in the notes. I said it very quickly, so hopefully somebody caught what I said. This is a hard one. Correct answer. Good job. People were actually awake. Good, good. That's good. Okay, last question. Aladdin, see if you can hold on to that. Which process is the most efficient at generating ATP molecules? Aerobic respiration, good job. Okay, let's see how the scoreboard comes out. Bronze, Rian. Silver, Shane. Aladdin, Aladdin, is it Aladdin? It is. Well done. Woohoo. Okay, so well done folks. We're just finished now. I have 2 quick things to say. One is, you know, you probably saw from this Kahoot, we have entered into more difficult material territory. You know, coming to the midterm, it's going to take a little bit of studying to really learn and understand some of this stuff. Point number 2 is that the midterm is 2 weeks from today, okay? So if you were somebody who did poorly on the first midterm, it's time to start thinking about looking at that material. And it's time to start asking questions and all of those kind of things, because I do want people to do better for the second midterm. We'll be doing some review next week. We have to get through photosynthesis and mitosis and meiosis first. So we have a little bit of material to cover. But I'm hoping to have lots of review sessions as well before the midterm. So that is all for today. Hopefully you have a great evening, and I will see you on Wednesday.